Hello, welcome to Stars, Cells, and God. My name is Jeff Zweerink. I'm joined in studio here today with Dr. Fuz Rana. We're going to be talking about AI, irrationality, and biochemical design. Fuz, good to have you in the studio today. I'm going to throw the floor over to you and just let you talk about your discovery on biochemical design.
Yeah, well, I'm going to start it off by asking you a question, Jeff.
Okay.
Kind of a pop culture question. Uh, and this will ease us into the discovery, but do you remember the song The Gambler by Kenny Rogers?
Oh, yeah. You got to know when to hold them, know when to fold them.
Yeah, that's right. Yeah. Well, that song was actually very much part of the cultural milieu in the late 1970s, early 1980s. Everywhere you went, you heard that song. And it was just a little bit of music trivia. It was written by a songwriter named Don Schlitz, and he actually wrote the song and made it available to Bobby Bare and Johnny Cash.
hmm
And they recorded the song, but it was Kenny Rogers' version that became the popular version.
Yeah.
And it was like a #1 country Billboard charting
hmm
single. And I think top in the Billboard Hot 100, it was like number 16 was the highest that went. So
Well, I know it was popular enough, not that I could do it now, but I actually could recite from memory all the verses and everything. So, I mean, it was a big song.
yeah, yeah,
And it was the Kenny Loggins or Kenny Rogers version.
yeah, yeah. So anyway, but as you pointed out, you got to know when to hold them, know when to fold them, which is, I guess, good advice if you gamble. But it's also something, interestingly enough, that cell membranes seem to be able to do, is that
Mm.
cell membranes are able to actually fold in order to maintain a constant surface area to volume ratio as cells grow or as cells differ in
hmm
size depending on the particular cell type. And that it turns out that this surface area to volume ratio is actually a very important geometric constraint that allows adequate flow of nutrients into the cell to maintain biochemical operations as the cell
hmm
either increases in size during cell growth or varies in size depending again on the cell type. So a research team from MIT just recently discovered that cells actively maintain a surface area so that the surface area to volume ratio is constant. And this is actually quite remarkable.
No, it is. I was because you just, I mean, that's one thing you work on many physics problems is like you figure out, does it go by the volume, by the radius, by the surface area?
Yeah.
Because if you, I'm presuming cells grow over time, and so the the surface area, the volume grows cubically. So as you increase the radius, it goes up by three r to the r cubed, whereas
Yeah.
the surface area goes up r squared.
Yeah.
So that means as the cell is growing, if it's going to maintain a surface to volume area, it's got to have a lot more structure in the surface area or something. Because I
Yes.
would I would assume it's kind
Yeah.
of it starts more dense and expands and gets less dense on the surface, if you will.
Yep.
But okay, so I see. This is exciting because I see where you're going. This has got to be a pretty complex feature to be able to do that
Yeah.
because it's real counterintuitive.
Yeah. Well, and kind of to bottom line it, and I have a slide that shows a picture of the journal article from Current Biology. Again, a team from MIT published this work just recently. But the bottom line is that this provides ongoing evidence that biochemical systems reflect the handiwork of a creator. That when you look at this particular system, it displays foresight. It's, as we'll see in a minute, it's going to require high levels of integrated complexity
hmm
with different biochemical systems. There is an elegance and an ingenuity to
hmm
the way this works, all of which are the types of words you would use to describe something that was designed.
Yeah.
Um, and from our creation model perspective, we believe that there is a creator who brought the universe into existence, and therefore we would expect to see evidence that the universe has a beginning, that there's design in the universe, but that design also extends to biochemical systems, to
hmm
living systems. And biochemical systems are the most fundamental systems of life. Therefore, in our model, we would expect to see evidence
Right.
for design. And this is really significant in the life sciences because many people operate in kind of in a framework that says, look, evolutionary processes can explain the origin and the design and the history of life, that biology in its totality can be explained through evolutionary processes. And if that's the case, then many people I've heard argue, well, a creator is
Hmm.
superfluous. What role is there for a creator? In fact, when I was an undergraduate student, I held that view myself.
Right.
And that made my, position of agnosticism very comfortable, right? And it was really in graduate school when I was confronted with the incredible design of biochemical systems that I realized there was a creator. But you can't really respond to the gospel until you're convinced that there's a creator that
Hmm.
exists and that creator is playing an active role in creation and an active role in human history. And
Hmm.
so this type of insight about design really is in many respects foundational to the gospel. It's not only an important part of our creation model, it's ultimately foundational to the gospel.
No, that's, I appreciate the way you're saying that because, just even as I've been reading through the Gospels and reading through Acts, kind of started a through the year and through at least the New Testament in a year reading plan. And one of the things that impresses me is how often the gospel, it's not like, oh, we're going to present the gospel. It's like, we're going to preach repentance. Well, that's an integral part of the gospel, but you can't repent of something you don't need to if you
Yeah.
if you feel like things are good, you don't need to repent for anything.
Yeah.
It kind of has this parallel to there.
Mhm.
If God doesn't exist, if he isn't active, why would I respond to him,
Yeah.
if you will?
Yeah.
So interesting parallel here.
Yeah.
So there's more to it than just saying here's the gospel. It's like there are things you have to believe in order to respond to the gospel.
Yeah. Yeah. That's really a really good point, Jeff. So, just a little bit of background information. We're going to get into some of the science behind this discovery here. And I got a picture here of a typical animal cell. And usually these are depicted as being spherical, right?
Hmm.
And then there's another slide here about the cell cycle. So, usually when a cell undergoes division, it winds up with two daughter cells that are roughly half the radius of the original mother cell. And
Okay.
then there's a growth phase where the cell grows in size and then eventually reaches a point where it will undergo again a process of cell division, which is a multi-step process.
Hmm.
But the point is that the size of a cell, if it's actively growing and dividing, is dynamic
Right.
throughout the cell cycle. Also, cells differ in size, and as you already pointed out, if you think of a cell as roughly being spherical, and that's not always the case,
Hmm.
right? That the equation for surface area is 4 pi r squared, and the volume is 4 pi r cubed, right? So if you
Hmm.
take the ratio of the surface area to the volume, you wind up having as the radius gets larger, the surface area drops, and it drops precipitously.
Right.
I think the equation is 3 over r,
Hmm.
right? So as r gets larger, because it's in the denominator, the
Right.
surface area rate the surface area to volume ratio plummets, and I got a slide showing that as well, just so people can visually see how the surface area drops. But the bottom line is that this is problematic
Hmm.
because if you don't have an adequate surface area, that is what's regulating the flux of material in the cell and out of the cell. So you're not going to get adequate nutrient flow and elimination of waste as
Hmm.
the cell gets larger and larger, right? And so what these researchers discovered is that there's an active mechanism in play that causes the cell membrane to fold so that it creates surface area through the folds that
Hmm.
allow it to maintain that constant surface area as the radius of the cell gets larger. Now,
Well, and that was the part, as I'm thinking through. Okay, so say you've got a cell that's 1 centimeter in radius. It's going to have a certain surface area to it. I could imagine that being the membrane being squashed up or compressed or something. So as it grows, you still have
Right.
you've got the same amount of stuff there, but you've got more volume that it can handle. But this is actually something very counterintuitive because instead of the membrane just kind of stretching and accommodating and getting spaced out, it's actually incre
Right.
dramatically increasing the amount because not only does it have to get larger to accommodate the
Right.
volume, it's got to get larger and have a bunch of structure in it.
Yeah.
That is really if I look and see if somebody built something that did that, that requires a lot of design. I mean, I see the design implications of
Yeah.
that right away.
Yeah. Well, yeah. And let's go back to the slide that I showed earlier about here's a typical animal cell. And as you're
Mhm.
pointing out, when you look at the cell membrane, it just looks like this flat,
Mhm.
nondescript envelope that separates the interior of the cell from the exterior environment. But when you blow it up and look at a small segment at a molecular scale, you see it's a very complex system.
Okay.
And it's out of that complexity that you're able to introduce these folds into the membrane. But the membrane consists of a bilayer, that's the matrix. And as the name implies, there are two layers that are joined together to form
Mhm.
this bilayer structure. And the matrix of each of those layers is formed to first approximation by molecules called phospholipids.
Okay.
And these molecules look like balls that have two long tails. And the tails are hydrophobic, they don't like water. The head group likes to interact with water. And so what happens is that in water, they spontaneously will form a bilayer structure where the phospholipids will associate laterally so
Mhm.
that the head groups form the surface of the bilayer on the outer and the inner monolayers, and the tails form the interior where they again aggregate laterally, and then the tail regions interact to
Okay.
form kind of a sandwich like
Right.
structure. And then embedded in the membrane or associated on the surfaces are proteins. Whole ensemble of different types of proteins are associated with cell
Mhm.
membranes. So when these researchers tried to determine what is the impact of cell growth on surface area, they couldn't just simply do it microscopically using microscopy, right? Because um, uh, you don't get enough resolution to see the folds, right? So what they
So how is that? Because I mean, you got your standard lab microscope. I know you can get down and at least see the cells, and it wouldn't
right,
surprise me that you can see the cell walls. I know we can do electron microscopy, which is looking at individual
right,
atoms. So I mean, it would seem we've got the
right.
range to do that. So how can we not do
Yeah,
that?
well, when you're visualizing cell membranes, you can either use a microscope, and that gives you very, it gives you crude architecture,
Okay.
and an electron microscope will give you the details of cell membranes, but you can't image the totality of the membrane at the level of an electron microscope. You can only
Okay. So
look at regions.
I can see things with the electron microscope, but I'm not seeing a large enough reason to see the structure in the membrane. So I'm
Right,
looking at
right,
just too small. Okay. I get what you're saying.
right. So the approach that this research team took, which I thought was ingenious, was simply to determine how much lipids and how much proteins were present
Okay.
in the cell membrane as the cells grew,
Mhm.
right? And so that's where that becomes a proxy for surface area. And I got a slide showing a plot of how the surface area increases
Mhm.
through folds as the cell volume gets larger. That's kind of the bottom line.
Okay.
And so this is again really interesting because it suggests it's a geometric constraint that the cell is actively managing. Now, one of the big questions the researchers have is how is the cell doing this? How does the
Yeah.
cell know how big it is? And then how does it know how to introduce the folds at the right amount in the cell membrane? Because if you're introducing too many folds, that doesn't hurt you in terms of flux
Mhm.
into the membrane, but it's a lot of energy
Right.
that's being spent to create materials that you don't need, right? So there's got to be an active way to monitor cell volume, surface area, and then to respond appropriately from a biochemical standpoint. They're not quite sure how the cell does this. That's going to be some very
Yeah.
interesting studies that follow to try to figure that part out. Now, what they suspect is there could very well be molecules in the membrane that are somehow sensing cell surface area and cell volume,
Hmm.
right? And so even though the overall composition scales as the cell gets larger, individual components don't. And so it could
Right.
be in some of those individual components where the signaling is taking place.
Yeah, that aspect of it is really interesting to me because it's like, as a human, I can look and say, "Oh, I can see how big it is. I can see what sort of," but there's nothing. There's no active mind looking at this.
Right.
And again, I think of the, like the Hoberman spheres, these things that can collapse down to this and expand out to this. It's like it's a pretty static structure, but the dynamic nature of this introduces a level of complexity and sophistication that is really fascinating.
Yeah.
So I would be interested to know in the future how what they figure out is
Yeah.
doing this.
Yeah. Well, and we actually have a pretty good understanding of how to introduce folds into membranes.
Hmm.
And I got a slide here from a review article that shows a number of different mechanisms. And bottom line is that there are certain proteins that will interact with either the surface or if they're embedded in membranes that will actually introduce folds.
Right.
But also the phospholipids themselves can be responsible for introducing folds.
Hmm.
If the overall geometric shape of a phospholipid is cylindrical, it'll form a flat bilayer.
Okay.
If it has a conical shape,
Hmm.
it'll actually introduce curvature as it packs.
Right.
And so by manipulating the phospholipid composition, even though, I mean, there's hundreds and hundreds of different species of phospholipids in cell membranes,
Okay.
differing in head group and in the fatty length and the degree of saturation of the fatty acid chain.
Hmm.
So by manipulating head group structure, by manipulating chain composition, you can create membranes that
Hmm.
are going to be more amenable to folding or membranes that are going to be less amenable to folding. So we have a general understanding of how to introduce folds into membranes, but specifically what's going on here, nobody knows. But
Yes.
to your point, it's such a dynamic system, and there's so many variables that you could manipulate and control. The level of control, the level of
Hmm.
and there's so many systems that are involved in producing and incorporating these molecules into membranes and monitoring their location in membranes and manipulating their location in membranes. You just have these highly integrated systems that are all working together to seamlessly introduce the folds at the right time in the cell cycle. This is absolutely remarkable in
Hmm.
terms of the foresight, the integrated complexity, and it's just an incredibly elegant and sophisticated system. This is the
Yeah.
type of thing that looks designed.
So, question I have: How in the what's the time scale on cell growth? So, yeah, I imagine there's quite a range in there, but you got some cells that are going to divide, grow, and then divide again. I mean, are we talking days, months, years,
It could be hours to days.
hours to days,
I mean, it
typically? So,
depends on the cells. Some cells will only replicate at certain times,
mhm.
in tissues but yeah, it's on the order of hours to days is typically what you're looking at.
Yeah, I guess it just that to me that says something about the mechanism and how well the mechanism that senses what needs to happen has got to. It's
Right.
like you could have things go on for a long time before you could sense out of balance. And if you only have a day or so where this is going on,
Yeah.
I mean, I guess molecular processes operating in hours and minutes is not the scale of it is just an interesting time scale to
Yeah.
me that you
Yeah.
can't have this long time to sense things. It's got to
Right.
be fairly quick. It sounds like that
Right.
it adjusts. So,
Well, and again, this is, clear evidence for design, but it does raise at least reasonable questions about how could a such a mechanism evolve? Because it's an either or situation. If you don't actively manage surface area, you're going to wind up with the cell not being able to sustain itself. So you don't have a lot
Right.
of leeway to develop kind of a stepwise system. It's almost as if the whole kit and caboodle has to come together all at once.
Well, so yeah, I was just trying to, okay, if I were looking at this from just a how might this come about, I could imagine a scenario where, there's got to be some range of surface area to density that works. It can't be just a number. There's got to be a range in there. And so I could imagine having this range where when things split or cells are small enough, when things split, the daughter cells are similar enough in size that it's within that range. And there's
Right.
just this small range to where, okay, so you can have replication without a lot of growth in there. And then at some point in time, there's, whatever the mutation that would allow more protein folding to happen. And so you get more protein. But it does, there's a part of me that says, okay, you look at the history of life on Earth and you've got single-celled organisms for the vast majority of time. It seems like there's quite a bit of time for something like this to come about.
Right. Yeah. Well, I'm not sure that this is a mechanism that applies to bacterial cells or archaeal cells.
Okay.
And or plant cells necessarily, because plant cells have a cell wall that's going to limit their size.
Okay.
But this seems to be applicable primarily to animal cells and is going to be probably most important in animal cells that are part of or cells that are part of complex multicellular organisms.
Mm-hmm.
So this kind of mechanism could most likely emerged 500 million years ago when animals first appear. It may have appeared even earlier in some single-celled organisms, but
Yeah.
but this is one of those scenarios where it's easy to maybe construct a just so story
Mm-hmm.
in evolutionary terms superficially, but when you start getting into what we suspect are going to be the molecular details,
Right.
that's where I think, to me, the suspicion arises: could natural processes evolve this kind of a system? Because
Yeah.
it's one thing to talk in general terms; it's another thing to talk in specific terms.
Mm-hmm.
And it's not just a single mutation here or there. It's going to be you suddenly are going to have to create a mechanism that produces only certain types of phospholipid species, and those are going to have to be properly distributed in the inner and the outer leaflet. And there's mechanisms, proteins that have to do that. They're called flipases and flopases and flip-flopases, believe it or not. That's, that are regular.
So the biochemists are as creative in their names as the astronomers are then. Okay.
Yeah. So when you start thinking about all what that has to happen, even though we don't know yet, but we know enough
Mm-hmm.
to know what this mechanism is likely to look like and how many biochemical systems are impacted
Yeah.
and have to respond appropriately, that's where I become suspicious. But
Well, and especially the other, that's what I typically find a lot of times. A new discovery will come out or something we find, oh, wow, we never saw this before. We've never seen this before. And it's like, there's a part where it seems to fit in, it just kind of fits naturally within the net. And then you dig into the details. It's like, oh, this is so much more sophisticated than I thought.
yeah,
This has that, we're just kind of beginning to see it. But the other part of this that stands out to me is that it's kind of it's not at the base of life, if you will, because single-celled life doesn't have this, but it's at the heart of multicellular life. If you want to have multicellular life, you got to have this mechanism in place. It's not like we can tinker around and play around with this and do it. It seems pretty essential from the get-go.
Yep.
And it's a very complex mechanism from the get-go.
Yep.
Those at least strike me as pointers to this being designed rather than just coming about by some sort of natural process, if you will.
Yeah. So there we go.
Well, cool. This is a fun discussion. I appreciate. I do have to say I don't. I've said this multiple times. I don't know that I'd want to do biochemistry, but I really enjoy talking biochemistry with you.
Yeah.
Because it's a cool field. So.
Yeah, it is. It's amazing. Well, what I've always liked about biochemistry is it's really an application of chemistry and
Hmm.
physics and
Right.
and so you're really applying different areas of science to living systems. So that's why it's fun and why it's, I think, broadly appealing to people,
Right.
and why a lot of people who are not formally trained in biochemistry end up working in
Hmm.
biochemistry.
I could see that. So, well, I'm gonna flip the discussion here, or at least go down a different train because I'll ask you a question. And actually, you'll probably remember this, but back when I first, within the first 5 years of working at RTB, you sent around an email that had this uh, paper generator. You put in a link in there, you just click on the link, put in a couple of keywords, generated this really long paper. Do you remember doing that?
I do.
Because I just remember looking at this paper and I don't even remember what it was because it absolutely made no sense. But it had you were reading it and it just had the flavor of, oh, this is actually a scientific paper. And then you looked at it and it was gibberish, if you will, but it had all the right phraseology, the right big words where you needed big words. It had terminology that seemed appropriate, and it was just fun to play with because it was like it was close to being scientific research, but it was just entirely hogwash.
Yeah.
And why I think that, or why I bring that up, is because what people have found as they're looking at AI, we've got these large language models, ChatGPT, Lambda, Llama, all these various different ones, is that the tone of this paper was, this is what's going on. I mean, there's nothing in the way it's delivered or in the cadence that indicates it's just entirely hogwash. And we see that same sort of phenomena with artificial intelligence, these large language models, is that they will give, or you type in a prompt, it'll give a result, and it's like, this is what's going on. And then, unless you know better, you could just believe something that's it's just made up. You got AIs have these hallucinations where they're saying things, but they're saying it so confidently, or it's the response is given so confidently that there's no indication that something's amiss or maybe to dig deeper. And this is not entirely surprising. I think I just like to bring that out because I don't know whether it's my era. I don't know how universal this is, but when I think of AI, I'm kind of thinking Spock.
Yeah.
Without any sort of, no emotions. It's just all rational. Everything is, if it says it, it's exactly right. If it doesn't say it, or if it won't say what's right. None of that stuff gets in the way. And if we approach AI that way, we're going to get burned a lot. And so I just want to kind of make people aware of the limitations that these things are out there. Because, in part because AIs are designed to engage you, not primarily to give accurate information, and so I think it's just always good to be aware of that. Really, what I found interesting in this discussion, though, was not that this is a problem of AIs, is that there are actually humans that have brain conditions that are similar to this. And there's a condition called Wernicke's aphasia, where people will, have had this. Often it happens after strokes. It's still trying to figure out what's going on in there, but where people will say, they'll just talk, and what's coming out from all the mannerisms looks like a coherent thought. It looks like, hey, I've thought about this, but it's gibberish in that it doesn't actually have any meaning, or it's incorrect meaning, but there's nothing in the delivery that tells you that. And what I found very fascinating about this particular paper, it's called The Comparison of Large Language Models with Aphasia, is they noticed that there's these similarities in there, and they wondered, is there a connection between these two? And specifically what they did is they were used functional magnetic resonance imaging, and it was resting state functional magnetic resonance imaging, to look at the brain states of people with this type of aphasia. And then they were able to apply an energy mapping, energy landscape tool to measure, uh, basically what it does is it goes in. It was originally used to measure magnetic moments of materials, but they're adapting this to look at the energy landscape of the brain,
Mm.
particularly looking at if you imagine you've got this kind of rolling plane, if you will, there are dips and divots and
Mm.
peaks and divots, and what they're looking for is characterizing how long does how long does it reside within a divot? How much does it transition between? And so looking at those two, the transition times and the dwell times, or transition frequency and the dwell times. And this particular model they were able to look at that with these aphasia paintings, but then they were also able to do that with the large language models. And what
Mm.
they found is that there were certain types of aphasia, there are different types of aphasia, and certain types that didn't have any parallels with,
Mm.
but certain ones, particularly this Wernicke's aphasia, had these parallels between these two
Mm.
models. And what I like and I think is cool about that is that normally the way to diagnose the aphasia is just by looking at the outputs of it. You watch somebody, you see. I mean, I've had interactions with various family members who've had dementia, and the way you tell it is you kind of start seeing the external signs. Well, by the time you start seeing the external signs, it's kind of far along, and it's hard to,
Mhm.
it's difficult to deal with. If there were a way to measure something before you could
Mhm.
see the external consequences, that would be cool. And it looks like there may be some potential, because what you can do is just look at functional magnetic resonance imaging, do this analysis on it, and by looking, there were a clustering where the people with aphasia, you could identify them
Mhm.
in that way. And so, they actually did that by looking at the large language models,
Mhm.
recognizing, hey, we can do this here, applying that
Mhm.
to the different types of aphasia, and says, hey, we may have, instead of waiting for the external appearances of the disease to show up, or the condition to show up, we might have a way to go in and
Mm.
just using a magnetic resonance imaging to be able to detect
Mm.
that it's going on, which, now that you have the capacity to do that from the state of the brain or how the brain is behaving, there may be ways to diagnose it, which may help us. If you can now go put that into the large language models, you might be able to develop.
Mm.
You can't experiment, or experimenting on humans is hard, but you can actually design
Mhm.
large language models that allow you to do various types of, well, what if we do this, or how can we do this, or
Mm.
would this solve it? To do all, kind of like using the AIs as lab rats or lab mice
Mhm.
to develop procedures or thoughts that might help us be able to diagnose and treat
Mhm.
these symptoms in people. And I'm just like, that's really cool. I'm skeptical that we're going to use AI well, but there are some really cool things that we're
Mhm.
going to be able to do with AI that can help people out. And
Yeah.
I just think that's fascinating.
Yeah. Yeah. Well, this is, I mean, really interesting work, because you could maybe argue superficially, well, does that mean that eventually AIs are going to be like us as humans if
Hmm.
there's that similarity between, different, you know, neurological diseases and the way large language models function? So how would you respond to that concern?
Yeah, I thought that was actually just a question I had as I was thinking about this discovery. So, if I'm saying, hey, we can use large language models in parallel to
Mm.
diagnose, so I think there's both an application and a limitation to that. And I would put it, it's like we do experiments on mice because they serve as a proxy for humans in a lot of ways. Now, nobody would claim, oh, mice are humans, but the way
Mm.
their biology works is similar enough that in certain instances you can use
Mm.
that to get insight into what's going on in the human body. And then there's still that
Mm.
final, okay, we got to actually interact with humans to do this. It wouldn't surprise me that a lot of the things we're doing that allows these large language models to produce coherent thoughts, or what we deem as coherent thoughts, that we're, in by and large, replicating a lot of what the human mind is doing from a biological or physiological basis. But to me, I think it's important to realize that distinction. So in AIs, we are developing, putting a whole bunch of silicon together from a hardware and a software perspective, and we're doing things that mimic human behavior. To the extent that mimicking is good, then we're going to be able to draw parallels. But I think there's, again, if Christianity is correct, one of the things I would argue is if Christianity is correct, yes, I am biological, but I'm not solely biological. So just because you've mapped out my brain and said, okay, here's the state, doesn't mean you've gotten Jeff Zweerink out of there. Jeff Zweerink is something more than just the brain states. And I think there's a lot of philosophical and theological, and even I would expect scientific research that shows that there's a distinction between those things. But in that, I would say to the extent you're replicating or approximating the biological, then yeah, you're going to get good insight. So it's kind of akin to what I was saying, with this discovery. When you first see it, it's like, oh, that I could see how naturalistically that would work, that it seems to lend credence to that. But then as you look into the details, it's like, okay, no, there's a lot more to that, where as we understand, the details are bigger. So initially I would say, oh, large language models, diseases, or, human conditions, there's a lot of similarity. Maybe these things are actually sentient.
Yeah.
And then as you look in more closely, it's like, no, there's components, but not the essential features.
Yeah.
That's kind of the argument I would make.
Well, because the idea of neural networks, which is one of the frameworks by which the large language models are implemented, right? That design of a neural network was inspired by the neural networks in the brain.
Exactly, right.
So the fact that you would see then some instances where the neural networks are behaving like the brain isn't necessarily a valid argument to say, hey, that these neural networks are just exactly like humans, or they're humans, but rather they're replicating it. And because of that, you now have these systems that, as you're pointing out really well, they just turn out to be useful in some instances because of that design similarity.
No, yeah, exactly. And, these are the sorts of things where, I've, like I said, I've had family members that have dementia. I know people who've had strokes. Yeah, there's lots of things that happen because we're biological beings who live in this world, and we get sick, and there are diseases. And I think, over the history, at least over the last 300 or 400, 500 years, our scientific advances have done a lot to help minimize the impact of those diseases, and also or conditions, and also give people capacity or ability to live more complete, more full lives. I want to be careful how you say that, because I don't want to imply that someone's got a disease is incomplete, but they can do things that are fulfilling and rewarding in more ways, and I think that's a good thing. I do think, that what comes with AI is that we're doing things that are remarkably close in the way the human brains, and we have to be, I think, because of the power that the tool has, we need to be careful how we use it so that we use it well, because anything, as Spider-Man, with great power comes great responsibility. I think these are very powerful tools. Let's be responsible so we use them well, but there are great things that are going to come out of this.
Well, I think AI is going to revolutionize science and the development of technology, probably even in ways that we can't even imagine, but we're getting a glimpse of this, and so I see a lot of the use of AI, at least from the scientific community, as being largely responsible, right? And there's a real desire to try to use it and use it well, which is encouraging to me. I think it may be in the more practical applications of it where things may run afoul, right? Where it's being incorporated into daily activities or being ceded control over certain things. That's where, to me, I think the real concern is.
Well, and I think, what gives me encouragement about that is that there are things that we can do, and whether this applies to everybody or whether it applies to Christians, I think it specifically applies to Christians because God calls us to grow and mature and put Him above all things. In everything, we're to submit to Him and order our lives to direct and glorify and honor Him. So when I'm watching ads on TV and they say, hey, using Gemini to compose a letter to somebody, I'm thinking, is that really what I want to do? Do I want an AI to be doing because a letter, if I get a letter from you, I'm expecting that it's thoughts you've had that are directed at me.
Yeah.
Do I really want to be doing things that make my relationship or use AI to make my relationship small? Well, no, I probably don't want to do that. But if I could use AI to make things faster so that I can do other stuff, or I could just be efficient so that I might be lazier. So all of that, it's ordered around, is this drawing me closer and helping me know God and love God and love people better? I think Christianity provides a great framework for doing that. And I want to encourage Christians, if we do that well, one, we're modeling how to do it, and that's going to draw people to Christianity and to the gospel as well. And so I think there's great opportunity here, although I'm a little anxious about it if I'm candid. So
this is something that I've given a lot of thought to that I find remarkable is that the ethical system and the wisdom system, if you will, the wisdom that flows out of Scripture is absolutely robust.
Yes.
We're confronted with advancing technologies and questions that the original authors of the Bible never could even imagine, right? But yet that ethical framework that flows out of what they wrote about, which we believe is divinely inspired, and the wisdom that flows out of Scripture actually equips us really well to engage these emerging issues that, are far beyond a pre-scientific context in which Scripture was delivered. And I'm not sure that there are other ethical systems that are based on some secular framework that actually can navigate these complex issues as well as the system of Christian ethics, because it's pro-technology and pro-science with the idea that these advances are going to promote human flourishing, help us to care for the environment. But there's also this recognition that humans are sinful, that we misuse those things that we develop, even if they're intended by some people for good, that we have hubris, that we lack wisdom, and we're overconfident, and at the same time, we respect human life and value human life. So we will say no to certain things if it undermines human value and dignity. So it's a great balancing between these tensions that emerge because of biotechnologies or things like AI technologies.
I think that's a great comment. Very insightful. I have found just the same. It's like, as I read through Scripture, there's an initial place where it's like, oh, wow, it looks like God's this, that, or the other. And then you realize it's like, oh, no, there's a depth. The deeper I've gotten in, it's like, I just remember reading through the Mosaic Law, and a lot of that, it's like, what does this mean? And then at some point in time, I recognized there's a whole lot of this where if I were on either side of the equation, I would say, yeah, that's a reasonable solution. It's like there's a depth to it that it at first glance it may seem troubling depending on what sort of cultural things I was bringing in, but it does reflect that God loves us, abundantly blesses us, recognizes that we are flawed and rebel against Him, and has put safeguards. It's a very robust system. I think that's just very well stated. So.
Yeah.
Well, thanks.
Fascinating stuff, Jeff.
It is. So, well, I want to just hope you have enjoyed your opportunity to be part of our dialogue. I want to encourage you to drop down in the discussion below, join the comments down there. We release new episodes of Star Cells and God each Wednesday here on YouTube and on your favorite podcast app. Want you to share this video with a friend and get into a good discussion with them. And remember, the more we know about science, the more reasons we have to believe in the God of the Bible.